Hello everyone, my name is Burak Gerçe and this is my presentation about nanosensors. I want to start this presentation with a question. What is nano? First of all, I want to explain when it's come. Nano means extremely small. It comes from the Greek word, nanos. They use this word for dwarfs. And we have another question. How small it is? It is 1 billionth of a meter. If you want to explain in real life, a strand of a human DNA is 2.5 nanometer in a diameter. Or it is about 1,000th of the size of the hair. We learn what is nano and when it's come. Let's start the talk about our main topic. Nanosensor. What are nanosensors? Nanosensors can be broadly classified into two groups. Those that have nanoscale dimensions and those that perform nanoscale measurements, but do not necessarily have nanoscale dimensions. Basically, the nanosensor is a sensor that either has to have a size in a nanoscale, or the distance between the nanosensors and the object it is. Observing has to be in the nanoscale, therefore a nanosensor does not have to be nanosize to be classified as a nanosensor. So, now we know what a nanosensor is, but why are they important? Nanosensors open opportunities to build new generation of devices. They are capable of interacting easily at the nano level and can observe unique process at the nano level, which are completely different from the macro level. Nanosensors have high sensitivity, which allows more accuracy, are small, durable, light and portable. Nanosensors have low power consumptions, require less sample volume for analyzing, and cause minimum disturbance to the observed material or process. They have low response time and more speed than other sensors, which allows them to do real-time analysis. It can detect multiple things at the same time, allowing for multiple functions. Because of all these things, nanosensors have a lot of potential in many fields. Now, let's look at the different classification of the nanosensors. Classifications of nanosensors. There are several classifications of nanosensors. Let's take a look at some of them. Active nanosensors require an external source of power in order to operate. Passive nanosensors uses external factors to do work. Absolute nanosensors. Absolute nanosensors have a set reference point. For example, an absolute pressure nanosensor measures pressure with reference to the zero pressure. Relative nanosensors. Relative nanosensors have a chosen reference point. Nanosensors are also classified according to the form of energy signal. They detect physical, chemical, or biological. All nanosensors fall under one of these three, but there are also more specific classifications based on the form of energy. Physical nanosensors. Physical nanosensors are used for measuring properties like temperature, pressure, flow, stress, strain, position, displacement, or force. Chemical nanosensors. Chemical sensors are meant for determining concentration or identity of a chemical substance. Biosensors. Biosensors are useful for dealing with biologically active substance. Nanobiosensors. Based on the interfacing of nanomaterials with biomolecules. Thermal nanosensors. Used to detect heat and change in heat. Optical nanosensors. They are capable of constantly monitoring chemical or biological process and are capable of converting the available information into signal useful for analytical purposes. They are considered better than other nanosensors for analytical purposes. Magnetic nanosensors used to detect magnetic fields and non-magnetic items inside a magnetic field. Now, we know what they are, why they are important, and some classifications, but how exactly do they work? Nanosensors work by converting the observed material or a process into electrical signals which can then be analyzed. Most nanosensors work by measuring electrical changes in the sensor material. For example, if a molecule of nitrogen dioxide comes in contact with a chemical carbon tube nanosensor, it will strip an electron from the nanotube, which will make the nanotube less conductive. The nanosensor will notice this and send electrical signals. Nanosensors detect the changes from the external interaction and communicate to other nanocomponents. Applications. Nanosensors can be chemical sensors or mechanical sensors. They are used to detect various chemicals in gases for pollution monitoring. For medical diagnostic purposes, either as blood-borne sensors or in lab-on-a-chip type devices. To monitor physical parameters such as temperature, displacement, and flow. As accelerometers in MEMS devices like airbag sensors. To monitor plant signaling and metabolism to understand plant biology. To study neurotransmitters in brain for understanding neurophysiology. Nanosensors aid in progression of fields such as medical technology, precision agriculture, urban farming, plant nanobionics, prognostics and diagnostics, source-based sensors, and many industrial applications. Nanosensors include carbon nanotube, quantum dot, DNA, peptide, all of them based fluorescent nanosensors, plasmon coupling, plasmon enhancing, magnetic resonance imaging, photoacoustic based nanosensors, all of them. Multimodal nanosensors, synergic nanosensors with multiple modalities to overcome individual challenges. Let's talk about first question. Can nanosensors warn of tsunamis? First of all, what is the tsunami warning system? The objective of warning system is to continuously monitor the state of the sea surface in near real time. This allows for a fast and efficient warning to be issued through the correct channels. A warning system consists of three components: scientific instrumentation, communication system, and warning system. First one, the scientific instrumentation is an assortment of sensors designed to detect various ocean activity, pressure, current velocity, and seismic sensors are the types of sensors used in an effective tsunami warning system. Some sensors target the tsunami wave over different ocean depths by measuring wave height. In order to do this effectively, the sensitivity of these sensors must be sufficient enough to detect changes in wave heights of a few decimeters over deep ocean. In addition, all other environmental factors affect the sea level, such as tides, atmospheric pressure, and wind, must be understood and monitored in order to separate these signals from the tsunami wave. The wavelength of the tsunami waves is different from the wind-driven and tidal waves. This allows for the identification of a tsunami by using spectral analysis, a process that identifies variation in cyclic components over a period of time. For example, ocean waves consist of different frequencies and amplitudes that are normally stable and predictable. Spectral analysis uses wave height versus time to determine the normal frequencies and identify changes or abnormalities. Once the observation of seafloor bottom pressure sensors, Global Navigation Satellite System (GNSS), and tide gauges or satellites have been collected, a fast transmission of the data to a processing center is required. For this purpose, radio transmission from a buoy to a land station is used. If the distance between the two antennas is greater than about 50 kilometers, a satellite link is established. The processing centers at the land station analyze the incoming data and detect potential sources of the tsunamis, and combine this with the received observations. The results are basis for the activation of a warning system. Now let's look at the early warning system. For example, German and Indonesian tsunami early warning system. The GTIFs consist of multiple sensors for comprehensive monitoring of seismic activity, water pressure, sea level, and meteorological data. The principal components of GTIFs are ground sensors, a global navigation satellite system buoy, and an ocean bottom pressure gauge. A space sensor is used as a radar altimeter. Communication systems are radio and satellite links from the sensors to a land-based tsunami warning system. We use these all systems, micro-electromechanical systems, ocean sensors. We can attach these devices, ships, buoys, rigs, anchors, automated under vehicles. If we sum all informations and attach the MM's ocean sensors, all required devices, we get the answer. Yes, we can. System MMs on anchor get the signal delivered to buoy MMs, then buoy MMs send the signal to satellites, and finally satellite gives us a warning signal, then we can protect ourselves. Nano-cantilever based biological sensors. Researchers at Purdue University have made a discovery about the behavior of tiny structures called nano-cantilevers that could be crucial in designing a new class of ultra-small sensors for detecting viruses, bacteria, and other pathogens. The nano-cantilevers, which resemble tiny diving boards made of silicone, could be used in future detectors because they vibrate at different frequencies when contaminants stick to them, revealing the preference of dangerous substance because of the nano-cantilever's minute size. It is more sensitive than larger devices, promising the development of advanced sensors that detect minute quantities of a contaminant to provide an early warning that a dangerous pathogen is present. The researchers were surprised to learn that cantilevers coated with antibodies to detect certain viruses attract different densities or quantity of antibodies per area. Depending on the size of cantilever, the devices are immersed into a liquid containing the antibodies to allow the proteins to stick to the cantilever surface. But instead of simply attracting more antibodies because they are longer, the longer cantilevers also contained a greater density of antibodies, which was very unexpected, said Rashid Bashir, a researcher at the Berg Nanotechnology Center and a professor of electrical and computer engineering and biomedical engineering at Purdue. The research also shows that the density is greater toward the free end of the cantilevers. The engineers found that the cantilever vibrates faster after the antibody attachment if the devices have about the same nanometer range thickness as the protein layer. Moreover, the longer the protein-coated nanocantilever, the faster the vibration, which could only be explained if the density of antibodies were to increase with increasing lengths, Bashir said. The research group also proved this hypothesis using optical measurements and then worked with Ashraf Alam, a researcher at the Berkeley Nanotechnology Center and professor of electrical and computer engineering, to develop a mathematical model that describes the behavior. The information will be essential to properly design future nanomechanical sensors that use cantilevers, Bashir said. Findings are detailed in research paper appearing online today in Proceeding of the National Academy of Sciences. The paper was authored by Amit K. Gupta, a former Purdue doctoral student working with Bashir and now a postdoctoral researcher at Harvard University. Pradeep R. Nair, a doctoral student in electrical and computer engineering. Demir Akın, research assistant professor of biomedical engineering. Michael Ledish, distinguished professor of agricultural and biological engineering with a joint appointment in the Weldon School of Biomedical Engineering. Stephen Broyles, a professor of biochemistry, Alam and Bashir. The work, funded by the National Industries of Health, is aimed at developing advanced sensor capable of detecting minute quantities of viruses, bacteria, and other contaminants in air and fluid by coating the capillaries with protein, including antibodies that attract the contaminants. Such sensors will have application in areas including environmental health monitoring in hospitals and homeland security. So-called lab-on-a-chip technologies could make it possible to replace bulky lab equipment with miniature sensors, saving time, energy, and materials. Thousands of cantilevers can be fabricated on a 1 square centimeter chip, Bashir said. The cantilevers studied in the recent work range in length from a few microns to tens of microns, or millionth of a meter, and are about 20 nanometer thick, which is also roughly the thickness of the antibody coating. A nanometer is a billionth of a meter, or approximately the length of 10 hydrogen atoms strung together. A cantilever naturally resonates or vibrates at a specific frequency, depending on its mass and mechanical properties. The mass changes when contaminants land on the device, causing them to vibrate at the different resonant frequency, which can be quickly detected. Because certain proteins attract only specific contaminants, the change in vibration frequency means a particular contaminant is present. Ordinarily, when using cantilevers that are on a thickness scale of microns or larger, attaching mass causes the resonant frequency to decrease, which is the opposite of what occurs with the nanoscale thickness cantilevers. Researchers believe the unexpected behavior is a result of the antibodies being about the same thickness as the ultra-thin nanocantilevers, meaning their vibration is more profoundly affected than a more massive cantilevers would be by the attachment of antibodies. The conclusion is that when the attached mass is as thick as the cantilever, then you not only affect the mass, but you also affect a K property called the net stiffness constant, and the resonant frequency can actually go up, Bashir said. Gupta measures the cantilevers vibration frequency using an instrument called a laser Doppler vibrometer, which detects changes in cantilever's velocity as it vibrates. The researchers then treated the antibodies with a fluorescent dye and took images of the proteins on the cantilever surface, proving that the density increasing with longer cantilevers. Nair and Alam then developed a mathematical model to explain why the density increases as the area of the cantilever rises. The model used a diffusion reaction equation to simulate the antibodies sticking to the cantilever surface. The research is based at the Berkeley Nanotechnology Center at the Discovery Park, the university's hub for interdisciplinary research. Our last topic, nanofabrication. Nanofabrication is the process through which nanosensors and other nanostructures are made. There are two approaches to nanofabrication. The first one is top-down fabrication. Top-down fabrication is a method that can be compared to sculpting from a block of stone. The base material is sculpted to the desired shape. The second one is bottom-up fabrication. Bottom-up fabrication can be compared to building a brick house, but instead of placing bricks one by one, atoms or molecules are placed one at a time to build a nanostructure. The most common top-down fabrication technique is nanolithography. In this process, the material required for the nanostructure is protected by a mask, and the exposed material is etched away. There are many different methods to etching. Depending upon the level of resolution required for the nanostructure, etching of the base materials can be done chemically using acids or mechanically using ultraviolet lights, X-rays or electron beams. There are also many other chemical and mechanical methods to etching. Most top-down fabrication methods involve etching. Have we any problems with nanofabrication? Currently, there are problems with both the top-down and bottom-up fabrication methods. The top-down fabrication method is more efficient and takes less time than the bottom-up fabrication method, but the equipment is expensive. The bottom-up fabrication method is cost-effective, but is not currently effective and takes much longer than the top-down fabrication method. Now, let's look at the ideas to improve this process. Ideas to improve nanofabrication. Many scientists are working on using the bottom-up fabrication method except making it self-assembling in order to make it efficient. But there are significant obstacles that need to be overcome. Other ways to improve the nanofabrication process in a smaller time scale include finding better materials for the nanostructures and combining certain element methods from the top-down fabrication method, the bottom-up fabrication method. These are my references. I used some PDF format documents, but I couldn't add those documents here. Thank you for listening to me. Stay home, be safe, be healthy.